Hello guys, welcome to Rebuild Academy. My name is Adil Momin and I welcome you to the sixth part of our series From Virus to Vaccine, Let's Fight Infodemic. I hope you all are doing well fighting pandemic as well as infodemic. But before we begin, let me tell you a very important point. It is extremely important and highly recommended to watch this series in order. Only then you will get a complete idea and a clear picture of every concept involved. The link is given in the description. Now let's begin. Till now we have talked about organisms, their types, then we learn about microorganisms and pathogens. We have also discussed cells, their structure and components, especially nucleic acids. Then we discussed health and diseases, infectious diseases and how they spread. And in the previous video, we have talked about the immune system. Right. Now today we will start our discussion specifically regarding viruses. Indeed, we have a basic idea about viruses from our previous discussions. Now let's dive deeper. In today's first topic, we will look at the basic structure of viruses and how they are classified accordingly. Then we will discuss the viral replication cycle. In this, we will learn how the viruses hijack and spread inside the host. It is important to note that the replication cycle is a part of the chain of infection which we have discussed earlier. The chain of infection explains how the pathogen spreads in the community, whereas the replication cycle explains how the pathogen spreads within the host. In the third topic, we will learn about variants and strains of the virus, how they are different and what changes occur in them. And in the end, we will discuss a specific example of one of the most common viruses, that is influenza virus, its structure, different variants and its replication cycle. Okay then, let's begin. Viruses are submicroscopic infectious agents. They can infect animals, plants and even microorganisms. We also learned that viruses are not living things. They are like robots. When they infect the host, they start replicating themselves. But when they are in inactive form, they are called virion. Where did viruses come from? There is a much debate among virologists about this question. Three main hypotheses have been articulated. The first one is the escape hypothesis, which states that viruses emerge from genetic elements that gain the ability to move between cells. This means they were initially a part of a cell but later got the ability to exit one cell and enter another cell. The reduction hypothesis asserts that viruses are leftovers of cellular organisms. This means initially viruses were possibly free living organisms that lost the genetic information over time as they adopted a parasitic approach to the replication. Whereas the virus first hypothesis states that viruses predate or co-evolved with their current cellular host. That is, they are on Earth from the beginning and are evolving along with other organisms. Viruses are so diverse in nature that it is hard to exactly know how they evolved. Now, viruses can be classified as enveloped or non-enveloped viruses. Every virus has a genome. These are the set of instructions for its survival and spread. And this genome is protected with the help of protein coat. If the virus has only this component, then it is called as non-enveloped virus. Enveloped viruses have an extra layer outside the protein coat, which helps them evade the immune response and facilitate virus entry into the host cells. There are proteins on the envelope called envelope proteins. They are responsible for receptor binding and fusion. We know that receptors are guards on the cells, so these envelope tricks them to allow access inside the cell. Consider envelope proteins as USB ports and cells as computers. They attach themselves to computers through receptors and start producing more viruses by using instructions in the virus genome. Now let's look at some characteristics of enveloped and non-enveloped viruses. Enveloped viruses are fragile because they are trained in a protective way and not used to harsh environmental conditions, unlike non-enveloped viruses, which are resistant and can tolerate harsh conditions. Therefore, enveloped viruses are very sensitive to heat, dryness, and disinfectants, and can be destroyed easily using sanitizers. Sanitizers rupture their outer protective layer, which results in the disintegration of the virus structure, and since there are no repair mechanisms in virus, they can't repair themselves. On the other hand, non-enveloped viruses are less sensitive to heat and disinfectants because their outer layer provides them more robust protection. The advantage that enveloped viruses have: they are difficult targets for the immune system because they have a special ability to evade innate immune response. On the other hand, non-enveloped viruses are easy target for the immune system. Another advantage that enveloped viruses have is they don't destroy the cell, called cell lysis, for their multiplication, whereas non-enveloped viruses cause cell lysis for their replication, which destroys the cell and causes a strong immune response. Rhinovirus and polioviruses are examples of non-enveloped viruses, whereas influenza virus and Ebola virus are examples of enveloped viruses. Viruses can also be classified as DNA viruses and RNA viruses based on types of genetic material. If the genetic material is DNA, then it is called a DNA virus. If genetic material is RNA, then it is called RNA virus. DNA viruses are mostly double-stranded as compared to RNA viruses, which are mostly single-stranded. Therefore, DNA viruses are more stable and have a structural advantage in the replication process. DNA viruses possess a large genome and are less dependent on host, while RNA viruses are of a small genome and hence highly dependent on the host. Let me put it this way: DNA viruses are like software and RNA viruses are like small set of programs. Therefore, DNA viruses are less dependent on the host operating system. On the other hand, RNA viruses, being a small group of programs, highly dependent on operating system for their execution. Right. The rate of replication is slow in case of DNA viruses. They have to travel to the nucleus of the host cell to start replication. On the other hand, RNA viruses can replicate outside the nucleus. The genes of the DNA virus have proofreading skills, which substantially reduces their chances of mutation. Hence, the ability to mutate, that is, mutability, is low in DNA viruses, whereas RNA viruses lack this ability, which makes them prone to mutations. Hence, they have high mutability. Now, let's start discussing viral replication. It is a set of steps through which enters the host cell and start replicating itself. The first step is called attachment. In this step, enveloped proteins interact with specific cell receptors and initiate the binding process. Consider this: you plug in USB to your PC. After this, penetration of the virus occurs through the fusion of the virus with the cell membrane and the virus enters the cell. In the following step, known as uncoating, the enzymes in the cell remove the envelope, which results in the release of the viral genome in the cell. Consider this as when you plug USB into PC, the operating system starts reading it. Once the viral genome is released in the cell, it hacks the cell machinery and starts producing proteins and the genome. This process differs for DNA and RNA viruses. In the case of DNA viruses, the viral genome has to travel to the nucleus of the host cell, but in case of RNA viruses, they directly attach to machinery and start production. Consider this: DNA virus is software which will attach to the operating system of the PC that is handling the protein machinery, whereas RNA viruses will directly attach to machines and start giving their own commands. Here also, you can understand why DNA viruses are less likely to produce faulty products because they are controlling from the nucleus. After the production, the components will be assembled. This process is known as packaging. The newly formed viruses are ready to live. Now they can leave the cell in two ways, either by rupturing the cell membrane or by gradually exiting it. The gradual exit doesn't cause the cell damage, thus don't spark a major immune response. On the other hand, cell lysis initiates a strong immune response, and these released viruses will start infecting other host cells, and the process will continue. But the immune system will also detect these activities and start immune response quickly. Now let's quickly go through all the steps. First, the virus will attach itself to the host cell and it will enter the host cell. Then through uncoating, the viral genome will be released in the cell. Then the synthesis of proteins and viral genome will begin, after which the assembly and packaging of all components manufactured will lead to the formation of new viruses, and these viruses will leave the cell either gradually or by cell lysis. This is the viral replication cycle. Now let's start our discussion regarding variants and strains. These two terms are often used interchangeably. We know that mutations happen all the time, more often in viruses because they don't have repair mechanisms like we humans have. So imagine a tree growing. As it grows with time, it starts dividing into different branches, and these branches further divide into smaller branches, and the process continues. Similarly, when a virus starts replicating, it goes through mutations, and these mutations produce a different version of itself. They are like branches, and these different versions also go through further mutations, which further creates different versions, and the process continues. Now, each of these different versions is called a variant of the original virus. So we can say that a variant is a virus with a slightly different genome. The most important thing about these mutations is that they are not deliberate. They are happening as a copying error. Now, what happens due to these changes in the genome? Well, that depends what change it brings in the set of instructions of the genome. This change can be positive or negative for the virus. For example, if the change decreases the ability of the virus to replicate quickly, it will retard its replicating speed. On the other hand, if this change increases its ability to bond more strongly with the cell receptor, then it will increase its chances to infect more quickly, right? So, the variant can be superior or inferior to the original virus. Now, these superior variants are called strains. Therefore, in the case of strains, the change in the genome has improved the ability of the virus. Scientists are constantly monitoring these variants through genome sequencing. If they notice some enhancement in the abilities of a particular variant, they call it variant of interest. And after research, if they confirm that variant of interest is actually more capable, then they classify it as variant of concern. It is important to note that not all variants are variants of concern. Now let's try to list some of the possible enhancements that a strain can get through mutations. It can get the ability to bind different cell receptors, like changes in the USB ports. It can bind more strongly to the same receptor. It can also start replicating more quickly in the cell or transmit more efficiently between hosts. The variant can also evade the immune response or get the ability to remain undetected in diagnostic tests. And it is also possible that few variants can reduce the effectiveness of the antibodies formed either from natural infection, from different variants, or vaccines. These all are just possibilities. Now let's talk about the influenza virus. It is the virus responsible for causing the disease influenza, commonly known as flu. Influenza virus is an enveloped RNA virus. There are 4 types of influenza viruses: type A, type B, type C, and type D. Type A influenza viruses can cause infection in many organisms, including humans, birds, and pigs, and they have high mutability, that is, ability to mutate fast. Type B influenza viruses are only found to infect humans. Type C influenza viruses are primarily found in humans, but they cause mild infections, and type D influenza viruses are primarily found in cattle. So, type A and type B influenza viruses are potentially harmful to humans, especially type A, which causes annual cases of flu. Let's discuss more about type A influenza viruses. Since influenza viruses are RNA viruses, therefore their genome is RNA, and they also have envelopes. They have two major types of surface proteins, neuraminidase, that is Na protein, and hemagglutinin, Ha protein. Both proteins are antigens, that is, they spark an immune response when detected by the body. N proteins are responsible for spreading virus particles among the cells, and there are 11 types of N proteins, named N1 to N11. H proteins are responsible for recognizing and binding to target cells. There are 18 types of H proteins, named H1 to H18. Now, the influenza A viruses are subclassified based on the combination of N and H proteins. They are strains of the influenza A virus. The most common strains are H1N1, also known as swine flu. It originated in pigs, but it spreads primarily from person to person. Swine flu made headlines in 2009 when it was first discovered in humans. And another strain is H5N1, known as bird flu. There is a total of 198 strains that are possible from 11 N proteins and 18 H proteins, out of which around 132 are found till now. Recently, China reported the first human case of H10N3 bird flu. Now you must be wondering how a single virus is able to take so many different forms. We have learned that viruses mutate, but let's go some deeper and try to understand what mechanisms are responsible for many strains. There are basically two mechanisms involved. The first is known as antigenic drift and another is antigenic shift. In antigenic drift, there are small changes in the genome that take place, which results in small changes in the structure and properties of HA and NA proteins on the surface of the influenza virus, and the new variants formed due to antigenic drift are closely related to each other because the changes are minor. This gives cross protection. That is, if you have immunity from the previous variant, then that immunity can provide you protection from this variant also. This protection can be partially, even not complete, but these small changes can accumulate over time and result in major change, which can give rise to a new strain with a significant difference from earlier variants. But it is important to note that even if the variants formed by antigenic drift are capable of evading the immune response, and the phenomenon of antigenic drift happens more frequently in viruses. On the other hand, if the mutation causes some major and abrupt change in genetic material, which results in significant changes in HA and NA proteins, then this is called an antigenic shift. The newly formed strain is totally different from the previous version. Therefore, there is no chance of cross protection in case of variants formed by antigenic shift. The new strain formed by antigenic shift is called as novel virus, and the immune system can't recognize this novel virus, but the phenomenon of antigenic shift happens less frequently compared to antigenic drift. Now, both the phenomenon of antigenic shift and antigenic drift is observed in the case of influenza A virus, but in the case of influenza B virus, only antigenic shift is observed. Since cross protection is available for strains formed due to antigenic drift, the vaccine made for the previous version of the virus can be reviewed and updated for the new strains. But in case of antigenic shift, the previous vaccine will most probably not effective and a new vaccine has to be made. So these were two mechanisms involved in the formation of strains of influenza viruses. it is important to note that not all viruses go through these mechanisms during their mutations. Especially, scientists believe coronaviruses are not prone to antigenic drift or antigenic shift. Now let's look into the viral replication cycle of influenza virus. The first step is attachment. The HA proteins bind with the glycolipid receptors on the cell and initiate the process, after which the fusion of virus and the cell membrane occurs, which results in the penetration of the virus into the cell. Then the enzymes do the job of uncoating and the viral genome is released in the cell. Since the influenza virus is an RNA virus, therefore it does not travel to the nucleus of the cell, but starts the synthesis of the genome and proteins outside the nucleus. Then the assembly of the genome and proteins is done by packaging, this resulting in the formation of new viruses, and these viruses leave the cell by gradual extrusion without cell lysis, and once the new viruses come outside, they start infecting neighboring cells. So this is a viral replication cycle of the influenza virus. This brings us to the end of today's discussion. Now as usual, let's quickly go through our today's discussion. We started today with a brief revision on viruses and also discussed some common hypotheses regarding their origin. Then we talked about enveloped and non-enveloped viruses, their comparison. We learned that enveloped viruses are fragile and sensitive to heat and disinfectants, but they are more difficult to target for the immune system, whereas non-enveloped viruses are resistant but easy pick for the immune system. We also discussed DNA and RNA viruses and learned that RNA viruses have high mutability and are capable of rapid replication, but also they are highly dependent on the host for their replication. Then we discussed the viral replication cycle, different stages in the cycle like attachment, penetration, uncoating, replication, assembly, and release. After which we learn about variants and strains and how all variants are not variant of concern and what makes them variants of concern. After which we discussed influenza virus, its 4 types A, B, C and D. We learned that the A and B types are more concerning for humans. Then we discussed the structure of the influenza A virus, in which we learn about HA and NA proteins, how their different combination can lead to different strains of the influenza virus. Then we discussed mutations, mechanisms in influenza viruses, antigenic drift and antigenic shift, in which we talked about how variants formed due to antigenic drift are related to each other and thus cross protection can be utilized in this case. Also, the previous vaccines can be updated and used, but accumulation of antigenic drifts can lead to antigenic shift, which give rise to the novel version of the virus, which is entirely different from the previous one and thus makes the previous vaccines ineffective, but antigenic shift are comparatively less frequent. And in the end, we discuss the viral replication cycle for the influenza virus. In the upcoming video, we will finally starting our discussion on coronaviruses and COVID-19. So, make sure you subscribe the channel and press the bell icon for timely notification. If you enjoyed and learned something from this video, then ensure that you will like the video and share it with others so that we all work together and battle the infodemic. If you have any suggestion, you can comment below. That's all for now. We will meet in the next video. Stay safe, stay connected.